So, hello everybody, I am Paula and she's Elena, and we are doing our thesis in the genetic department, in the University of Barcelona. And our supervisors are Florenci Serras and Montserrat Corominas. So today we want to talk about a model to study regeneration that are the fruit flies. Now, okay, so the first evidence about regeneration was done in the ancient Greece. So Greek mythology tells us the legend of Prometheus. Prometheus was a mortal that stole fire from the gods. When Zeus knew it, punished him. And the punishment consists on sending an eagle to eat his liver during the night. But surprisingly, the day before, when Zeus arrived, saw that the liver was completely regenerated. So Zeus started to think that Prometheus was immortal. And as you know, all the legends have a true part. And here the truth is that in human body, the liver is the organ with the high regenerative capacity. But in humans, not only can regenerate the liver, because there is a continuous renewal of blue cells, skin, and mucous membranes. In this kind of cells, there is a balance between production and destruction. Also, we have partial regeneration when we break a bone or a muscle. And also, when we cut the skin or we suffer a burn or something like that, skin can regenerate, but not completely, because scars appear. So in summary, humans have limited regeneration capacity.
However, if we have limited regenerative capacity, there are many cute animals in the nature that have the intrinsic skill of regeneration. And for example, I'm pretty sure that if I ask you to have your hands, many of you will have cut the tail of a lizard when he was a child and then have seen that the tail was able to regenerate. But apart from the lizards, there are many animals with the intrinsic skill of regeneration. For example, the simple hydra, the amazing axolotl, or the beautiful starfish. And we need the requirement of a model to study regeneration because we can cut ourselves in pieces to check what's happening in us. So from all of them, our favorite model is Drosophila melanogaster. And maybe you're wondering why, and the main reason is because it's really cheap to work with it compared to another modern animals. But apart from that, it's easy to work because you can have a new generation of flies in only 10 days, and because of that, the experiments are so fast to do. Also, there is a 60% of the genome of Drosophila shared with humans, so we can extrapolate our results to humans. And finally, there are advanced sophisticated genetic tools that allow us to generate mutants in order to study the genes important in this process.
Okay, so to familiarize you with flies, you have to take a look on the left side of the slide, okay? This is the life cycle of Drosophila melanogaster. So when a male and a female fly meet and like each other, they do, and the female fly puts thousands of eggs. So these eggs grow and become larvae, and then these larvae enters to metamorphosis to give rise to adult flies. So where is the regenerative capacity in the flies? So it's during the larval period. So in the larvae, there are many epithelial structures called imaginal discs. Okay, so these imaginal discs have the intrinsic skill to regenerate after different injuries. So specifically, we work with one imaginal disc that looks like that under the scope. So as an example of how a disc regenerates, you can see in this video a magnification of this group of cells in the center of the disc. So as you can see, there is here a big hole that is the injury. Okay? The cells that are surrounding the wound are approximating each other to close the wound. Okay, so, and the green cells are the ones that can proliferate and recover lost tissue. So the regenerating disc looks like that under the scope.
Okay, so our main goal is to know what's happening at a cellular level, and thanks of many bioimaging tools, now we are able to know it. So if we look inside the cell, when there is an injury, there are many proteins that get active because there's a signal that goes inside the cell, and then these proteins can get active. This activation will lead to the activation of another proteins, like in a cascade of activation. And finally, there will be one protein that will enter into the nucleus and then will bind the DNA. Once that is attached to the DNA, this protein will produce some marks that will be the instructions that will say, hey gene, time to your function, get up. And then this gene will lead to the transcription and will form the RNA. And this RNA will lead to the formation of new proteins. And those proteins will be the ones that will allow regeneration. So our aims are to know which is the signal, how this can go inside the cell, which are the proteins that are active, which are the proteins that are attached to the DNA. We also want to know which are those marks. And finally, we want to know which are the genes that are being transcribed in order to generate new proteins. So as an example, if you cut a disc like it's shown here in the slide, you can see that the disc is able to regenerate in the video. But the interesting thing in this video is that there are two proteins, one expressed in red and the other in green, that are being expressed at the same time that the disc is closing and healing. And these are two very important proteins for this process. So in our lab, what we are doing is to study which proteins are being active and are being transcribed using tools like that. So to conclude, our main goals are to figure out which genes are involved in regeneration, to understand the gene network beyond this process, to find out how chromatin changes can trigger gene expression, and finally, to extrapolate the results to mammals in order to cure or to find some treatment for this illness that need the growth of some tissues like cancer or something like that. And finally, we want to thank the organizer and thank you for your attention, and also to our lab mates for being always there. And that's all.